this is the 9th day of online faculty development program. Participants, he is Shailesh sir, and he is going to take the second part of experimental designs and their importance. Thank you, sir.
Okay. Good afternoon to all the participants. Now, therefore, yesterday we have discussed the background of the experimental design and certain aspects. What are the factors? We have also discussed the various terminology which is used in any experiments, and on the basis of this, we have also discussed the importance of accuracy. We have also discussed the importance of precision and what is the importance of both the things, accuracies and precisions, that we have discussed. Today we are discussing about the experimental design and some of the analysis. Any one of you have any questions, you can ask me. Either you are not following in between lecture also. So now first step of the experimental design. So what are the various steps of the design? So first step of the experiment design is the planning phase. And unfortunately, we all, teachers as well as students, they are giving less importance to the planning phase. Second phase is the execution phase or actual experimental phase. We are giving maximum time. Of course, it's required the time, but we are giving more time as compared to what we should give for the execution phase. And the third one is the analysis phase. Now, once again, we'll come to the first planning phase. Now, what includes in a planning phase? So it includes, you have to state the problem, you have to define the objectives. So first of all, you have to find out the title of your research work, what you want to do, or title of the experiment, what you want to do. And on the basis of this, you have to define the objective of this. Then select the output response variable. That when you are doing this particular experiment, then what types of the result you will get, that we have to find out. Select the input factors to be explored. What are the factors we want to study during the experiment, that we have to study. Select the factor level. At what level various factors are to be studied, that we have to decide. And then you have to go for the design of experiment. And you have to see that what response you want, what type of the factor you want to study, what is your problem, what are your objective. On the basis of this, we have to think about the design. Or if you have in your mind the type of the design you want to study, then you have to find out that what are the previous 5 factors which are suitable to design the experiment, what you have thought in the mind. Then determine the sample size, how much samples require, what sample is required. Determine the number of the replications, what you have to do, and experimental plan. So this phase, normally we tell the student that you go for the literature survey, and on the basis of the literature survey, you design the experiments. Many students, they say, we will do the literature survey throughout our research work of 3 or 5 years. At many occasions, even teachers are not very clear, or the researchers are not very clear regarding what type of the results they want, and for obtaining that result, what type of the experiment design is suitable. So they just select a topic, and after selecting the topic, immediately they will go for the XYZ design. As I have seen nowadays, since last 10-15 years, if you check any research thesis, then RSM, response surface methodology, that is the commonly used by most of the students, by most of the research laboratory, many times without understanding the importance of that particular statistical design. It may be possible that whatever the experiments we are doing, that may be done by much simpler experimental design as compared to the RSM or composite design or hexagonal design, or there are much more complicated design. But as nowadays all these designs are available online, so if you go to your laptop and if you download this, designs are available. You put your parameters, immediately they will give the designs, and once you do the experiment without knowing why that experiment are needed, what are the consequences of doing all this experiment, you will get some results. And the online itself, they will analyze your result and they will give you a long list of the interpretations. And I have seen that in many theses, these interpretations are verbatimly copied from that design software and they will be given in the thesis. So this planning phase, now there are three phases: planning phase, then actual experimental phase, and the analysis phase. Normally it is thought that it should be 4 Gen 2 Gen 4 or 4 Gen 2 Gen 3. Means 4 time is required for planning phase as compared to the actual experiments. Double the time is required. So if you are doing your research for 3 years, then it's required a very long period for planning your phase. Not necessarily that during that time you don't do any experiments. But first you select your problem, you discuss your problem very critically, and then find out the objectives of that particular problems. And then you have to go for the execution phase. Same way, in case of after obtaining the results, many times, being a teacher for 35 years and guided more than 35 PhD students, I know, on the just 2 or 3 days before submission of the thesis, they will complete the final experiment. So we don't get enough time to analyze. So that is also not good. Because if you are not properly analyzing the results, you put your results in the thesis, in the research paper, but it is not the 100% your effort or your hard work you have done that you are representing in terms of the analysis. If some other person who is knowing and who is giving them more time, he can get much more better conclusions from this experiment. So execution phase, that is actually the work you are carrying out. But the first phase and the analysis is important time. In case of the execution phase, normally we are conducting experiment and collecting the data. I have also observed that many of the theses, they are having the bulk of data, huge data. And out of data, the number of the conclusion they have made, they are very non-significant. Because the proper time was not given, proper first phase was also not decided, and due to this, people are not able to conclude properly. As what we have discussed in a previous lecture, the badly designed experiment. A good design experiment or correct design experiment will give you the better analysis, better conclusion, and significant conclusions. Now, in analysis, what we are doing, we are identifying the significant factors and their interactions. We fit and finalize the modeling equations. Being a biology student, we are afraid of the mathematics, and modeling equation will come. Now, many times we think that why we want to spend time. But once you decide the modeling equations, then it gives the concept. If you change any of the parameter, what could be the predicted results? Now, when I was doing my PhD, and we have calculated these modeling equations. Now, if you start line to line, then it was 2.5 page long equation was calculated. Because I was having the backing of the engineering college, and with the help of this my guide, he was metallurgical engineer, and from that I have learned in 1977-78. I have learned that what is the importance of a proper experimental design. At that time, this computer and online calculation was not available. I have calculated the result nearly for 21 days. Every day I was sitting 10 to 11 hours just by taking a calculator, normal calculator, and I was calculating and the modeling equation was solved. But due to this, now the equation was so perfect, so in many industries, many of this parameter and equation is used for the extraction of metal ions. Evaluate the conclusions for the impact on the current process and downstream customers. Conduct the cost benefit of your experiment or implications. Consider the next generation design of experiment and optimizations. So, as we said that after deciding the title of the research or title of the experiment, what you want to do, then we have to propose the hypothesis. Of course, if it is your research is a discovery type of the research, then hypothesis may not be necessary if it is a discovery. But that is rarely happens. Most of the time, more than 80-90% of the research, they are the hypothesis-based research. So, hypothesis, what is hypothesis? Hypotheses are a proposed solution or solutions of the problem on an ad hoc basis in terms of a statements are known as a hypothesis. So what statement you are doing in the hypothesis is the solutions or what outcome you are going to get. That is on ad hoc basis. That either you have to prove or disprove the hypothesis. You need not to worry if you are able to not able to prove the hypothesis. If it is disproved, then it is also equally important. And you have to say, now this is not possible. Whatever the hypothesis I have put forward, now it may not be happening under these experimental conditions or under the type of the samples what I have collected. Null hypothesis, which denote as a H0, it means there is no difference between the two groups or two sets of experiment, or there is no difference between any of the observed value and sample value and the population value. So whatever the difference observed are due to purely fluctuation in the sampling, or it is by chance method, which is denoted as H. So suppose if I am doing experiment, if I am testing, being a microbiologist, if I say E. coli strain A1 and E. coli strain A2, so I am saying that both the strains are similar. There is no difference in terms of the glucose utilization of this. Or suppose we can say nowadays very recently in UK, the new strain of the COVID virus is found. New strain of COVID virus is found. Now, so as a null hypothesis means this new strain is not having any difference in terms of the infectivity, in terms of the spreading of this disease. That is null hypothesis. If by data, by analyzing data, if you prove no, this old strain and the new strain, the difference is significant, then we can say the null hypothesis is rejected and we have to go for the alternative hypothesis. So H0 is a null hypothesis, H1 is an alternative. So in H0, mu minus mu2 means a mean value of the one set and mu value of the second set, whatever set, is 0. Or we can say mu1 is equal to mu2, where mu1 and mu2 are the mean of the two populations. So this indicates that it is no difference between this. H0 may usually be considered the septic hypothesis, nothing new or interesting happening there. And that is why normally as a researcher, we want to reject the null hypothesis, as we want to prove that we have done something and this is our contributions. So to show our contributions, normally we are more and more interested to reject the null hypothesis, and then we are going for the alternative hypothesis, which is denoted as H1 or HA. Alternative hypothesis is a negation of a null hypothesis. It is expressed as H1, where mu1 is not equal to mu2. Mu1 is not equal to mu2, means two populations or two experiments what you have performed, one may be wild type, one may be a mutant type, one may be effect of pH, one may be effect of temperatures, then they are not equal. There is some significant difference, and that is alternative hypothesis. So HA or H1 may usually be considered the researcher's hypothesis, but it is very easy to prove or disprove the null hypothesis but it is not prove or disprove the alternative hypothesis. So generally that's why we are selecting the null hypothesis. If we say the null hypothesis is not acceptable, when null hypothesis is not acceptable, it means we may accept the alternative hypothesis. Testing or verification is more objective. In case of null hypothesis, it is very easy and more confidently testifying. Testing is more consistent with the probability models, which is not happening in the alternative hypothesis. But when these three things are not matching, then you can say we are accepting the alternative hypothesis, or at that particular significant point, we are not able to accept the null hypothesis. When we are doing this, any hypothesis testing, normally we know that we go for 95% significance or 99% significance, and where the possibility of the two types of error. Error 1, that is known as type 1 error, a conclusion that a factor produces significant effect on a response variable when in fact its effect is negligible. So that is first error. When the effect is negligible, then also we are considering that factor producing significant effects. It is a one error, or we can say a false alarm. While in type 2, a conclusion that a factor does not produce a significant effects on a response variable when in fact it is affecting meaningfully or significantly. It means suppose you are giving one medicine to the patient, and if that medicine is effective and you consider that this medicine is not effective, it means you are making the type 2 error. In first one, even a medicine is not effective and you consider that medicine is effective without considering the significance, that is type 1 error. So these type of error are playing very significant crucial role, and to balance, because if you are going to more and more strict with a minimum type of the type 1 error, it means you are increasing more and more type 2 error, and vice versa. If you're giving liberty to the type 1 error, then type 2 error will be reduced. So in both the way, we should be very careful, and that is why normally in case of any good design experiment, 95% significance or p-value is 0.05, 99% significance where the p-value is 0.01, or 99.9% significance means p-value is 0.001. These are the three different stages are considered. But under normal experiments, p 0.05 or 95% significance is more than sufficient to predict any significant error. This we already discussed. Now this is how it works. Reject one, then other is selected. You're selecting other, the other is failed. Level of significance that we have discussed. Normally we have to go for 0.01 or 0.05. Mostly we are collecting. Now initial level we are deciding here. Then we have gone for this, why it is happening like this. This is also we have discussed. Now, as half of the time of this lecture, nearly 20 minutes we have spent for this. Now we will go to the experimental design. Normally in case of experimental design, if you want to bifurcate them, there will be two different ways. One is informal experimental design and second one is formal experimental design. Now, some of you will think, or you will may tell me after the lecture or before the lecture is over, sir, this we are doing now, what to discuss, what is the control design and what is the control after the test and before the test and all these things. But this is my experience of these years, and I found that many times we are doing this type of mistake. And here I want to tell you that once you do this mistake, now you cannot conclude your result properly. So there are two different group of the experimental design, informal design and formal design. In case of informal design, before and after without control. Before and after without control. Second one, after only with control. And third one, before and after with control. Have you thought about this three possibility? If you have thought about, then you know what type of the mistakes we are doing. If you have not thought about, then the mistake will continue during your process. Now, this is, as we are having only 1 hour and I want to give the explanations of all the possible designs. So I will be going little bit quickly. Before and after without control. It means what is happening in this type of experiment. Test area. This is the test area. Level of phenomena. You are giving the treatments and level of the phenomena you are measuring. Correct? So, and this is normally we are doing. This is normally we are doing. So, effect of treatment, we are calculating x minus y. So, this is level of phenomena. What is the, suppose if I want to measure the amount of the glucose or amount of the amylase activity or activity of the any other enzymes or the microorganisms, I will see what is 0 hours result and what is final hours result. And then I will do x minus y minus x. Y is the final hours result, x is the 0 hours result. And we consider that our experiment is over. Now, most of the people are doing this. They say, yes, we have taken 0 hours result. And why to use the other controls? Even now, just I want to share with 1 or 2 minutes what interaction I had before 30 years with my MSc student. We are going for the alcohol fermentations. And I told that you have to measure the sugar concentrations in an uninoculated medium and inoculated medium both. And then at the end of second day or third day, whatever amount of the substrate utilized or alcohol produced, they have to measure. One group of students, 1 or 2 students, they argued. Now we know that we have added 17% of the jaggery. Why you are forcing us to do the estimations? I have tried to convince them. They are not convinced. They say, no, we will not do. I tried to give all the explanations. They were not ready. And that matter was extended up to so that it has gone to the head of the department. Head of the department also said, no, they know that they have added 70% of the jaggery. Why you are insisting that how much jaggery are added? We'll take jaggery. Then I told, sir, see, these are the problem. Suppose if I purchase jaggery from A shop, jaggery from the B shop, jaggery from C shop, do you think that all the jaggery having the same amount of the sugar? If it is not there, then no, we know that we have added 17% of jaggery, but how much amount of the measurable sugar is present, that is essential. Now, I will not go into that story, but it was difficult time for me to convince even other teachers, and that students, no, they want to continue. They have gone for 1 or 2 days strike. I have to take it. I said, no, this is scientifically wrong. The second incidents happen when we are doing the biochemical test. Those who are microbiologist teachers, they know we are adding the Andras indicators in a fermentation tubes. So one of the fellows saying, no, we will add after the incubations. I say, no, you have to add before sterilizations and you have to sterilize after addition of these reagents and then you have to inoculate. No, that also the argument, but that you will come to know when what is happening. So here, before and after without control. This is, you are taking reading before control, before the experiment and after, but you are not keeping any control. So this type of the problem will come. In second experiment, after only with a control design. So here you are giving the treatment introduced. So you are getting the result, level of the phenomena after this, and you are putting the control. You are putting the control. And no, this is also not true. What I have explained this. No, you have kept control. You have kept control. But both the analysis you have done in a control after the treatment. We don't know what is happening in the 0 hours. What is happening in 0 hours that we don't know. So that create a problem. So we have to go for the third one, very simple one. before and after with control. Before and after with control. So this is a control experiment where we are taking the reading before we start the experiment, where we are taking the experiment, then we do the treatment, we take the reading, that is Y, before experiment is X. In control, we are putting the control flask without inoculum or without enzyme or whatever it may be, the treatment means it is absence of a treatment. And we take the results before starting experiment. We continuously put that control flask 2 days, 3 days, 4 days, whatever days we are keeping the test flask. And then we are taking the reading. And that is A and Z. So a real, your outcome of the result is X minus Y, sorry, Y minus X minus Z minus A. That is the real outcome. If you are not doing this experiment like this, then you are missing something, some data, and that will affect your final result. That will affect your final result. So, but many of these experiments, they are the one factor at a time experiments. These are the one factor at a time experiments. And in this one factor at a time experiments, now interaction of the factors we don't know. Interaction of the factor we don't know. Then some other, what we have discussed day before yesterday, the confounding factors, experimental error, residuals. Residuals means a difference between a predicted value and obtained value. All these things, which is this, whatever the difference, what we are getting, that is due to what? Either it is due to the independent factor what you have studied, or it is independent factor plus some of the factors what you don't know how they are affecting. Many times when you are doing the field experiments, there are so many factors which we are not noticing. Even if you notice, you cannot control them. So you have to nullify them. You have to, well, the factors which we are not able to control, what we are not able to analyze, then we have to nullify them. And for nullification of this, you require a specific type of the experiment. So this type of experiment we are discussing in the upcoming slides. So a simplest experiment is completely randomized layout. Completely randomized layout. Are you familiar with this type of experiments or not? If not familiar, then you can ask me in between also. In case of completely randomized experiment, what is happening? Suppose if I want to study A, B, and C treatment. Suppose if I want to study A, B, and C treatments, and as we know that normally minimum 3 replication is must. So suppose if I want to do the 7 replications of A treatment, B treatment, and C treatment, this is not a factorial experiment. This is a individual experiment A, B, and C. But to get more acceptable results, to minimize the error, we are doing the replication. We are doing the replication. Now, suppose, now this is my experimental setup. This is my experimental setup. So, if I am testing 3 different parameters, and these 3 parameters, each parameter, if I am replicating 7 times, it means I am doing 21 experiments. Is it clear? If many times what happens, people will consider this 1, 2, 3, 4, 5, 6, and 7 is a A treatment, 8 to 14 B treatment, and 15 to 21 C treatments. So what wrong we are doing here? And if I perform experiment, this is first, second, third, like this, then I will go to the 14, 15, 18, 20, and 21 in a sequence. It's a wrong way. It's a wrong way. Why it's a wrong way? Now suppose whatever this this blue color, light blue color, and till light blue color, that indicate some inherent difference between these 3 particular things. If I am doing the experiment of A factor in a dark blue color, then whatever the difference between a first and the second blue color, no, that is constantly it will giving more results in a 1 to 7 experiments. 8 to 14 experiments, it gives less results. And in 15 to 21, till it gives the less results. This would be anything. This would be anything. This would be anything. It may be the self of your incubator. It may be the place of your incubator shakers. Either you are putting all the 7 flasks in the center of the incubator shaker, and the second 7 flasks, the B flask in the periphery of this, and third the 7 flasks on the tail periphery. Then there would be some difference in a temperature, in a flow of air, in a shaking condition, and many things that will affect. Suppose if you are putting all the flasks in a topmost cell, center cell, and a bottom cell, then also there would be difference. In agriculture, suppose no, this would be the top soil on one of the end, and this dark blue. Then this is the medium soil, and this is on the other end. We don't know, because soil, no, it may be possible that the dark blue may be having more amount of the salinity, or a lowest one that is very, very here the red blue color may be having more amount of the fertilizers. So this heterogeneity is there. So in case of completely randomized experiment, you are not dividing the soil when it is not possible, possible to do all the further divisions. Then what we have to do, that randomly 7 experiment of A, we can say A1, A2, A3, A4, A6, A7, B1, B2, B3, B7, C1, C2, C7. Then you have to give 1 to 21 number in this experiments. And then one by one you have to select. Suppose if I get the first cheat, which is C1, then I have to put the C1 here. Then second cheat, suppose it's C5, then I have to go for C5 here. Suppose third cheat, maybe A5, then I have to put A5 here. Suppose my 11th cheat, maybe C6, then I have to put C6 here. The 12th one, maybe A2, then A2 plus here. So randomly I am putting the flask, randomly I am performing the experiment, randomly I am selecting the place of the experiment, randomly I am giving this to the 21 student. So I am not giving that A treatment to the girls only, B treatment to the only boys, like not that. So it is randomly mixed. So whatever the difference of a particular plot, or whatever the difference of a particular gender of the student, either the boy or girl, or suppose I am using a reagents, sigma reagents, and we can say high media reagents. So I can divide some of the treatment would be done with a high media, some of the treatment with the sigma, but it is not that only A treatment will done with a high media and B or C will done with a sigma. So this will minimize the effect of any reagent, effect of any glassware, or effect of any student, or effect of any block. But here we are doing randomly. So no other factors would be considered here. No other factors. But till as we are doing the replication, and replications are repeated at many places, it will give the better results. So here I have shown now this how the ABC. So if you will find here 1, 2, 3 and 4. 4A come in a upper. Here 2A are come here and 1A. So if I find, because all other things are identical except the heterogeneity of this. So if the results of this and this, it should give the identical similar results. But effect of this and this is different. It means these particular things, what we can say, is having either beneficial or harmful effect as compared to this. Similar way, B is repeated here, B is repeated here, B is repeated here. So in all the three treatments are repeated, and within a treatment, any difference, then difference is due to the first line, second line, and third line. So that what we have said just now in previous slides, that control is also before the experiment, after experiment, and treatment, and all these things would be nullified. So this is the simplest experiment which can take care of some of the factors, Second experiment, which is randomized block layout. Previous one was completely randomized design. Here this is randomized block layout. Here now see this dark blue, light blue, much light blue, and this. Here I have divided, in the previous experiment I have not divided them. Here I am dividing as a block 1, block 2, block 3 and block 4. So here what I have to see, that if I am doing A, B, C and D 4 treatment, then all these treatment should replicate all. Here I am doing 5 treatment, so all the 5 treatment should replicate in each block. It is not that A should not come here or B will not come here. So in case of randomized block layout, what is happening, that all the treatments are replicated in all the blocks. So suppose now if I want to have a 5 treatment with a 4 replications, then this is the replication. So E 1, 2, 3 and 4. Can you see? 1, 2, 3 and 4. E has replicated 4 times, but not either in the same column, not in the same row. I am not taking E here. I am not taking E here. So any heterogeneity either in this form or in this form will be taken care by this type of the experimental layout. Similar way, B is here, B is here, B is here, and B is here. So this is also representing all the 4 blocks at a various angle, either in the beginning or at the center or middle. So similarly, if you check this, then you will find treatment A or B or C or D or E. All the 5 treatments are represented in each block. So a number of the treatments, so replications, if you are going for any treatment with an X number of replications, then you require X number of a block. If a number of treatment, if you are going for a Y replication, then you require Y number of the plot. So this is which is requirement before you start your experiment. Now this the design will say now how I am performing this experiments. So randomized block, suppose if I want 7 treatment A, B, C, D, E, F, G, and I am having only the 3 different type of the block, then I can replicate it for 3 times. So this one is example of randomized design. This is also an example of randomized design. Then randomized block designs, 3 treatment, A, B, C, and 7 replications, that is shown here. Latin square. So in case of randomized block designs, it take care of this one, but it will not take care of this and this. So if I want to take care of a horizontal and vertical heterogeneity of the experiment, I have to think about the Latin square layout. So in case of Latin square layout, the number of replications should be equal to the number of treatment. In case of randomized block design and completely randomized design, there is no necessity of the number of replication and treatment. But when you want to go for Latin square design, so your plot should be divided in a square, or the number of experiments, depending the treatment and replication should be the same. So here what is happening, in previous randomized block design, I have shown you that each treatment has replicated, repeated here, here, or here. Like here, each treatment. Now 5 treatments. So E. So E I am finding here. E I am finding here. E I am finding here. E I am finding here, and E I am finding here. So can you see this horizontal line in all the horizontal line E has come. In all the vertical line also column also the E has come. In a previous one it is not necessary. In a previous one it is not necessary. In case of Latin square, so there is any difference, any heterogeneity in this or this will be taken care. So if I take the E here, and if I take the E here, it will give the idea about the any heterogeneity in the columns. If I will take result here and here, it will give the difference between this as well as difference between the row. Here also difference between the row and column. So Latin square designs will give more control regarding the horizontal and vertical differences. In case of lab you are performing, then it will give the whatever the 2 types of the heterogeneity what you are thinking about, that will be taken care in this type of experiment. Now the third experiment, that is the factorial design. In case of all the previous experiment, you know the effect of a factor. You may be knowing the effect of the level, but we are not knowing the effect of interactions. We are not knowing the effect of interactions. And that is very, very essential. Because in case of biological science or in case of the chemical science, when you are studying more than one factors, definitely that factors may have some interactions. Either the interaction may be beneficial or interaction may be harmful. So to understand the interactions between factor A and B, or factor B and C, or factor A, B and C to all the 3 gathers, or suppose 5 factors, then how the A is interacting with B, how A is interacting with C, how A is interacting with D, and how is A is interacting with E. Similarly, how B is interacting with C, B is interacting with D, B is interacting with E. Third one, how C is interacting with D and C is interacting with E. And last one, how D is interacting with E. So this is the design. This is the way it's designed. I am giving you the simple experiment, simple 2 factors if I want to study. 2 factor if I want to study, suppose pH and temperature, and 2 level of pH and 2 level of temperatures. So number of experiment which is required to perform in this is 2 by 2. 4 experiment I have to perform. 4 experiment I have to perform. That will give the idea of interaction A with B at 1 level and 2nd level, and B with 1 level and 2nd level. As for example, a treatment A. As you can see treatment A, as you can see treatment B. The experiment variables and level 1 and level 2. So level 1 we can say low level, and level 2 suppose we can say high level. So in this first what I am studying is treatment A is low level, treatment B is low level. In this experiment I am studying treatment A is low level, treatment B is high level. In this experiment I am studying the treatment A is high level, treatment B is high level. In third experiment I will study the treatment B at high level and treatment A at low level. In fourth I will study treatment A at high level and treatment B at low level. So suppose in cell 1, so here A is represented. It means, what does it mean? Here I am doing this experiment with a low level of A and high level of B. Low level of A and high level of B. Sorry, high level of A and low level of B. And here high level of B and low level of A. Here I am studying high level of A, low level of B. Like this, factorial design, you can go for the combinations. And then if only the simplest factorial design, you can go for minimum 3 replications. So the same 4 experiment you have to replicate for three times and then find out the mean value, find out the standard deviations and calculate the significance factors. Now suppose based on this, if I want to study 3 factors, suppose pH, temperature and salinity at each of the 3 factors at 2 different levels. How many experiments are required? How many factors, how many experiment treatments are required? If I want to study 2 factors at 3 factors at 2 levels, how many experiments are required? So if I want to study 2 factors, 3 factors at 2 level, 8 experiments are required. 8 experiments are required. So from this a simple formula, suppose if I want to do 3 experiment, 3 factor at 3 level, how many 27 experiments are required? 27 experiments are required. If I want to do 4 factor at 2 level, 16 experiments are required. If I want to study 5 factor at 2 different level, 64 experiments are required. So this will give you the idea how many factor you want to study and at what level you want to study on the basis of experiments, number of experiments you know. Now suppose if I want to study A, B and C, and what are the 8 combinations of this A, B and C. I want to study at a 2 different level, we can say level high and level low. So how I have to write this experiment, design of experiment? I have to make the first column, I have to write 0. The second row of the first column, capital A, small a. Then the second row of the first column, small b. Third row of the first column, small a, small b. Fourth one, now AB has come, so I have to say C. Then next one curve, AC, BC and ABC. Have you got the idea how I am making this number of experiment? So my first is 0. So neither A, B or C is appeared. It means I have to study all the 3 factors at low level. So, and when in the second row of a first column A has appeared, it means I have to study A factor at high level and B and C factor at low level. In next row, first column A and B. So I have to study A factor at high level, B factor at high level and C factor at low level. Then fourth experiment C. So A and B are absent. So A factor at low level, B factor at low level and C factor at high level. And the last experiment, 8th experiment, where in the 8th line of the first column, I am writing A, B, C. It means A at high level, B at high level and C at high level. So if this is the way you have to design the experiment, When you are designing this experiment, that is known as full factorial design. But when you want to study more factor and at more level, it is because, see, suppose if you want to study 5 factor 2 is 64 experiment. But if you want to study a 4 factor at 3 level, then 3 into 3 into 3 into 3. Very high number of experiment, So that is why now partial factorial designs are also available. If you go to any computer and if you write this, you want to study a 3 factors at 4 level or 4 factors at 3 level, immediately they will give you the partial design. And computer will select that you need not to go for all the factor analysis. They will give you around 20, 25, depending upon the design you are selecting, this many experiment. And if you analyze this experiment, you can predict the value of a 64 experiments. So this is a partial factorial design. And after doing the partial factorial design, you can go for path of specification. Other is the hexagonal design, composite design, as I said, RSM, which is used by many students and many laboratory very frequently. What are the main uses of a factorial design? It provides to analyze the interaction between the factors. Any factors, what is that interaction that you come to know, which is not possible in any other experiments. And it is very essential to know the interactions to come to the conclusion. It provides equivalent accuracy. It is giving high accuracy. When now there are procedures, suppose if you are doing 2 raised to 4 or 2 raised to 5 experiments, then they will say just you select any one. So out of suppose 2 raised to 4, 16 experiment or 2 raised to 5, 32 experiments, you select any one point. Suppose I have selected ACD. It means now this experiment I have to repeat for 6 times. Instead of repeating all the 16 or 34 experiments for 3 times, I select, that is also randomly you have to select any one point, and by using the randomized table or just by closing your eyes and putting your finger from a bulk of this sheet you have prepared, and that experiment would be repeated 6 times, and that will give you all the benefits of replicating all the 32 or 16 experiments 3 times or 5 times. So this is a scientifically proved that it gives nearly more than 99% accurate reading as compared to completely replicating the design. It permits various other comparisons of interest, and that you can find out. Difference between the experimental and non-experimental research. In case of experimental research, we are able to control and manipulate the environments of the research, including the predictable values which can be changed. Or on the other hand, in non-experimental research, we cannot control or manipulate the researchers at will, and it is based on the survey. So many confounding factors, many factors which is beyond our control, that may be also responsible. Suppose if I want to know now what is the spread of the coronavirus in Ahmedabad. If I do the experimental research, I can perform this experiment in a laboratory where I can control many of the factors. But here, if I am analyzing the data, I don't know how the people are staying in a Naurangpura area, how the people are staying in a Bapunagar area, how the people are staying in a, we can say, Zoapura area, or how the people are staying in a Wadaj area. So the factors from which group of the family the patient is coming, either higher income group, lower income group, either educated, uneducated, what is the age difference. So this is a non-experimental research where a survey is done. While if I want to do the same experiment in the laboratory, then I can select, yes, I will give this vaccine to 25 to 30 years of age, 35 to 50 years of age. I will give male this much, female this much. If I will give people coming from a rich family this much, people coming from lower middle class family this much, people coming more slum area from this, then you will get many factors which you are controlled. And these people are kept under the control conditions. Type of the food you are providing, the way either they are smoking or not smoking, that everything will control. Problem of non-experimental research is it is more difficult to conclude. The relationship between the cause and effect cannot be established. No, it is just prediction. The correct effect will not be established. Independent variables are not introduced or withdrawn or manipulated. In case of non-experimental research, independent variables are not controlled, not introduced, or not manipulated. These are the difficulty of this. Experimental research is suitable for the research whose goal is to examine cause and effect relationship. And normally, as being a people of science, we want to find out, examine the effect of cause and effect, and relationship between these two. Use of experimental research may be in the physical science, chemical science, biological science, in medicine, social science, education science, psychology, human behavior, and many more fields. No, due to the shortage of time, I may not give you the explanation of all this. But if you have any questions regarding this, what brief idea I have given it on the experimental design and analysis, then I want to ask something to any participants. Do you think that knowing these experimental designs are useful to you to go for any new experiments? I am audible to you?
Yes, sir.
Okay, okay. So any questions or anything? See, I have to finish this. Normally 4 or 5 lectures are required to explain this.
Yeah.
I have to finish in 1 or 2 lectures. Normally, a factorial experiments I require 2 lectures. A 1 hour, 2 lectures minimum for the factorial design to give you the brief idea. Similar way, the Latin square will also require 2 lectures, because how to calculate, what to find out the residual value, how to find out the significance of this. But here we have to give you the brief idea about the experimental design. I think
તમે જે ક્વેશ્ચન્સ પૂછો તો એટલે કહું છું. અમારા જેવા જે લોકો યુજીમાં જે અત્યાર સુધી ભણાવે છે વર્ષોથી, ખાસ કરીને પીજી નો કે અનુભવ નથી, એટલે આ જે તમે એક્સપેરિમેન્ટ આઈડિયાની જે ડિઝાઇન ને ખાસ કરીને પ્લાનિંગ કઈ રીતના કરવું એ ખરેખર બહુ ઉપયોગી છે અમારા માટે.
અને અંડર ગ્રેજ્યુએટમાં પણ તમે જે એક્સપેરિમેન્ટ કરો છો ને એમાં થોડુંક જ મોડિફિકેશન કરવાની જરૂર હોય છે. એ સહેજ જ મોડિફિકેશન કરો ને તો મેં જોયું છે કે તમે સહેજ મોડિફિકેશન કરશો તો તમને બેનિફિટ શું છે કે તમારા ક્લાસમાં 20, 25, 30, 40 સ્ટુડન્ટ હોય છે, એટલે તમને રેપ્લિકેશન મળી જાય તો તમારા વર્ષે 1 or 2 સારા સાયન્ટિફિક પેપર નીકળશે. તમે જે એક્સપેરિમેન્ટ તમારા રૂટીન લેબોરેટરીમાં આપો છો ને TYBSCમાં કે એમાંથી ડેફિનેટલી જો તમે આને થોડું ફોલો કરીને કોઈ પણ એક્સપેરિમેન્ટ કરાવશો, સપોઝ એઝ ફોર એક્ઝામ્પલ those who are having microbiology, suppose gram staining, તો તમે એમાં સેફ્રેનિન કેટલું નાખ્યું, જન્સન વાયોલેટ કેટલું નાખ્યું, જન્સન વાયોલેટ કરતાં આ બીજું કોઈ સ્ટેન નાખ્યું. સપોઝ ઇફ યુ આર કમિંગ ફ્રોમ બાયોકેમિસ્ટ્રી એન્ડ એ તમારા જે પણ કેમિકલ્સ છે, સપોઝ યુ આર પરચેઝિંગ ફ્રોમ સિગ્મા, યુ આર પરચેઝિંગ ફ્રોમ હાઈ મીડિયા, યુ આર પરચેઝિંગ LR ગ્રેડ, યુ આર પરચેઝિંગ GR ગ્રેડ ઓર AR ગ્રેડ, એ કેમિકલ નાખીને તમે કરો અથવા તો કોઈ કંપનીના, definitely your result would be very useful and a company તમારી પાસે આવશે. I have given this type of experiment. You know this AquaGuard, those who are producing this the water suppliers. No, I have designed all the experiments for the AquaGuard and all the model they have developed. No, initially I helped them to develop their 5-6 models, even that UV control, UV radiations, and just giving to the MSc student. Only you also have the less time to work because you are having more workload. But આમાંથી કોઈ પણ તમે જો ડિઝાઇન કરીને એક્સપેરિમેન્ટ આપો ને તો રિઝલ્ટ તમને બહુ સારા મળે. And I think my first slide is showing my email address. So if you have any questions, anything at any time you can send me, or at any time if you require my help for designing experiments. Definitely in microbiology I will be more helpful, but even if you are coming from botany, zoology or biochemistry or a biotechnology, I will be helpful to give you the design of experiment.